Joining us today is Fraser McCready, who is a PhD student in molecular genetics at the University of Toronto. Fraser was actually my TA this semester in my neurobiology class, and during our tutorials, he told us about some research that he was doing at his lab in this past year, which was published in Nature Neuroscience. So congrats, Fraser, on the publication, and welcome to the show.
Awesome. Yeah, thanks for the introduction.
Maybe we can start off by talking about this paper that you guys recently put out there. The title is SHANK2 Mutations Associated with Autism Spectrum Disorder Cause Hyperconnectivity of Human Neurons. Why don't you tell us about what you guys found? What was your role in this publication?
So, I guess you can tell kind of by the title of the paper, our lab mainly studies autism spectrum disorders, but we study them by using what are called induced pluripotent stem cells. And we use those kind of as a model system for studying these disorders. So I guess essentially how this sort of disease modeling system works is that what we can do is take, I guess, easy to access cell types such as skin cells or blood cells from a blood sample or a skin sample, and kind of wind the developmental clock backwards a bit on those cells. To reprogram them back into pluripotent stem cells. And the useful thing about stem cells for us is that we can kind of turn those stem cells into different types of cells in the body that we're interested in studying. So once we have those stem cell cultures, what we can do is kind of start to wind that clock forward again. But this time, kind of nudge those cells down a different developmental trajectory so that they become brain cells like cortical neurons instead of like the skin cells that they were originally reprogrammed from. So kind of the goal of this paper was to reprogram these IPS cell lines from individuals who have autism that we know is associated with this, with a mutation in this gene called SHANK2, which is known to be associated with autism, and it's kind of this big scaffolding protein that functions at synapses where neurons communicate. And so the goal was to I guess, make these IPS cell lines, differentiate those into neurons, then kind of study how these neurons are growing and connecting and communicating differently in a dish. So that's something I guess we'd call cellular phenotyping. So I guess when I actually joined the lab, I was taking over this project from a previous graduate student named Kirill, who's the first author on that paper, who was just about to finish. And he had actually had all this phenotyping stuff done already, right? So when I joined the lab, this paper had actually already been submitted for peer review. And when I sat down with James Ellis, my PI, about potential projects, he was saying, yeah, we have this cool phenotype. We know that these neurons essentially make more synaptic connections with each other. They grow larger, their dendrites are more kind of branched and complex, and they seem to be receiving more excitatory inputs. So what your project could be is basically starting to dive into what's going on at the molecular level in terms of molecular mechanisms that could actually be causing these neurons to be overconnected, right? I was like, yeah, that sounds really cool. And then we got our peer review comments back from the paper that Kirill had submitted. And, kind of the consensus among the reviewers was, among some other things, that this is a really cool phenotype. You guys have done a lot of work with this. But in order to publish it, we want you to tell us some sort of molecular mechanism, right? And so it was like, well, that was going to be my 6-year PhD project, but I guess we'll try to do this as quickly as we can, right? To try and find out like something, right? So my main role in this paper was to analyze a lot of RNA sequencing data that we had collected for some of these mutant cells and the controls, and kind of do some analysis to figure out, well, what is it that in terms of all the different genes that are being expressed in the mutant cells compared to cells that were made from unaffected individuals, how are they being expressed differently, right? And so normally when we do this sort of experiment with say different types of cells, you kind of end up with a list of like maybe 100 or so different genes that are being expressed differently. But when we did this for the SHANK2 cells, I think we actually found it was somewhere close to like 3,000 or 2,000 different genes that were expressed differently, which is like this crazy amount. So what I ended up doing was kind of following up on that and trying to see, okay, well, obviously we're not going to be able to pick out a single gene here that's the main root of this problem that we're seeing. But what we can try to do is organize these genes into groups based on their function, right? And then try to find, well, are there any sort of biological functions that seem to be overrepresented in terms of the number of genes they have that are expressed differently. And so what we ended up finding was that there were a lot of genes that are related to processes like synapse assembly, genes related to processes like sort of neurite growth and development, as well as just glutamate receptor signaling. So the type of signaling that you would expect to be important in like excitatory activity. We also found that there were genes related to processes like synaptic plasticity that were changed as well. So we kind of presented that as a bit of a mechanism in terms of, this supports the phenotyping that we did that shows that these cells are overconnected. And then to try and, I guess, do a bit of a rescue, one of the other things that the reviewers had asked us to do was, there had been some other papers that were published on mutations in a gene called SHANK3, which is related to SHANK2. And they had found some drug compounds they could put on these cells that would kind of rescue the phenotype that they're seeing. And so they said, well, why don't you try putting these on your own cells and seeing if that rescues the phenotype as well. So Kirill and myself, as well as an undergraduate student in our lab, did a lot of work with kind of this mini drug screen we did, where we put these different compounds on, as well as kind of an additional drug that I thought was maybe interesting to look at. And it's actually kind of funny because when Kirill and I were kind of talking about like what kind of drugs we want to include in this screen that we're doing, I had suggested this compound called DHBG. Kirill's kind of response was, yeah, we should put this on. We'll see what happens, but likely it's not going to do anything. But like let's just try it and see what happens. And so we did this sort of drug screen experiment, and it turned out at the end like the only compound that seemed to actually rescue the behavior of these cells was this DHBG compound. The sort of main findings that came out of this paper were, one, the fact that these neurons were overconnected was very novel because in a lot of research that's been done on autism spectrum, usually the type of phenotype you find is that the cells are underconnected. They make less synapses, they're smaller, they're less active. And so, us finding this was interesting. The mechanism stuff that came out of it, where we found that, things related to synaptic plasticity were changing, was sort of interesting for us in terms of where we could go with future directions and trying to follow up more on what's going on. And then the drug screen results, where we found that this DHBG compound, which is a drug that activates a glutamate receptor called mGluR5, the fact that treating these cells with this drug compound was able to rescue some of the overconnectivity phenotypes we were seeing, also kind of pointed us in a direction towards narrowing in on a more specific mechanism that could be causing this overconnectivity.
I did have a question about something interesting you mentioned. So you said that usually you would expect like maybe 100 genes to be changed, either upregulated or downregulated compared to normal. But you said that you guys found 2,000 genes had a difference. So do you guys have any like speculations or like insights into why that might be? Because from what I understand, SHANK2, like the protein, is not a transcription factor. It's not something that causes the transcription.
Exactly. Yeah. Mm-hmm. Yeah. So like, the 100 gene number is just kind of like a rough estimate. Like, it obviously depends on what type of mutation you're looking at when you do this RNA sequencing. Well, as to why we might be seeing so many different genes changed, I think my kind of hypothesis about that is that the actual function of SHANK2 at the synapse is to spatially organize a lot of different signaling cascades, right? So there's a lot of different signaling events that are sort of initiated by either things like that synapse receiving some kind of excitatory activity, or different growth factors binding, etc. And a lot of the times what ends up happening through those signaling cascades is you get signaling back to the nucleus that initiates some sort of transcriptional response to that initial input. Especially when you think about the fact that one of the phenotypes that we've seen with these cells is that they make more synapses, right? So you can think either you're seeing differences because the signaling pathway itself is being affected, or maybe you're just getting more signaling to the nucleus because there's more synapses. We haven't really investigated that any further at the moment, but that was sort of one of the things we were thinking about. And I think what you said about it, the fact that this isn't a transcription factor, I think is really important because that was kind of our response too, right? Like the other disorder that we study in our lab is called Rett syndrome, which is associated with a mutation in a gene that's supposed to be a global transcriptional regulator. And when we do RNA-Seq on those cells, like that's what we find is that, about 100, maybe 200 genes that are differentially expressed depending on how you do the analysis. So yeah, the fact that, this heterozygous mutation in just a scaffolding protein can cause such a massive disruption of the transcriptome is really interesting to us.
I guess that just speaks to, in addition to what you guys find out about a possible connection to autism, this is also important in that it shows us the effects of synaptic plasticity, like what happens when you have synaptic signaling and what happens when something different in that whole mixture can cause a change in the entire neuron in so much of the transcriptome.
It's really fascinating to me to look at all the different signaling pathways that function just at the synapse and how they all kind of crosstalk with one another. I spent a lot of time specifically at the beginning of my PhD when James had kind of tasked me with, okay, your project's trying to figure out what's going on in terms of molecular mechanisms. The first thing I wanted to jump into was, okay, well what's actually going on at the synapse? And I feel like my desk in the lab kind of looked like some crazy conspiracy theorist at some time where it just got all these crazy signaling pathways drawn out and trying to figure out how everything's related to one another. And at a certain point you kind of have to give up on that and try to focus in on something more specific because there's just so much going on that it really is incredible to think about the kind of processing that goes on just at individual synapses themselves.
Everything is so complex in the biology of people, so it must be hard to really hone in on one thing to focus on at a time.
Yeah, and I think it's you're absolutely right about that, and I think it kind of, to me, emphasizes the importance of collaboration in research these days. Like the more we learn about things, the kind of the bigger the wealth of information there is that you need to kind of know if you want to really have a good handle on what's going on. And at a certain point, it just becomes too large for any one person to be able to tackle on their own, right? So we all kind of have to specialize into becoming experts in a very small piece of this puzzle, and then collaborate and talk with one another to try and, I guess, integrate all those ideas into a more complete picture of what's going on. So yeah, I think collaboration has become very, very important in research these days.